So we are looking at 16.5 enzymes, and enzymes are proteins. So before we get there, I wanted to just make a point on 16.4 where we discussed denaturation of protein. On page 569, there is another chemistry to health link on the denaturation of protein in the mad cow disease. You can see how a normal protein, mostly alpha helix, and the abnormal protein in the beta plated sheet, that is another example of denaturation of protein. But we are going back now to 16.5 enzymes. What are enzymes? Enzymes are biological catalysts, and they are needed for most chemical reactions that take place in the body. So in this, we are going to look at the role of enzyme and name them and the reactions. So what is an enzyme? Enzymes are biological catalysts. They're proteins, and they're needed for most reactions. Like I said, here you can see there is a reaction without a catalyst, and it needs energy, higher energy than the same reaction with a catalyst, which can be accomplished at a lower energy. So what this catalyst is doing is increasing the reaction rate by decreasing the energy needed, increasing the reaction rate by decreasing the energy needed, and itself not participating in the reaction. So in this particular reaction, we are seeing the enzyme carbonic anhydrase. It is lowering the activation energy of this particular reaction going on, where the enzyme carbonic anhydrase catalyzes the rapid reaction of carbon dioxide and water to bicarbonate and hydrogen ion. And due to the presence of this particular enzyme, the reaction takes place about 1 million molecules of carbon dioxide in 1 second. That's how fast the reaction happens due to the presence of the enzyme. So what are enzymes again? They are biological catalysts that increase the rate of reaction but decreases the amount of energy needed to take the reaction forward, and itself is not part of the reaction. So the name of the enzymes are derived by replacing the end of the name of the reaction or the reacting compound with the suffix ase. Sucrase catalyzes the reaction of sucrose. Describes the compound or the reaction that is catalyzed. You can also oxidase, oxidation reaction, oxidase. It catalyzes oxidation reactions, oxidase. It catalyzes the reaction of sucrose, sucrase. So the reaction or the component that is going through the reaction, so you can have both of those as your basis of your name, or pepsin, trypsin. These are digestion enzymes. So some of the names have these bases. So again, here we have a classification, types of reactions. Oxidation reactions are called oxidoreductases, transferases, transfer group of atoms, hydrolases, hydrolysis, lyases, isomerases, ligases. These are all names based on the type of reaction that the enzymes, these are the class of enzymes. These are the type of reactions they catalyze. So we can connect the classes to the type of reaction. There are some 6 of these main classes. Okay? Classification, study check. So now we are going to use what we heard to see what's going on. Match hydrolases. So I'm going to say hydrolysis reaction. Transferases, transfer a group between 2 compounds. Isomerases, rearrange atoms in a molecule to form an isomer. Ligases, form bonds between molecules using ATP energy. So that's what we discussed. Excuse me. How to name an enzyme based on the reaction that it catalyzes. So we are naming enzyme classes, not specific enzyme, but enzyme classes. So this is what we did. Now, name the classification with what, how does this work? The mechanism is what we are looking at. So all enzymes are globular proteins. Globular proteins are tertiary structures. They are large structures. In this enzyme molecule, this entire enzyme molecule, there is an active site. There is an active site. And that is where this region, active site, this holds the substrate. Substrate is the reacting molecule. How does an active site hold on to the substrate? There are specific R groups interact with the functional group of the substrate to form hydrogen bond, salt bridges, or hydrophobic interactions. So the active site has R groups. The active site has amino acids. This is a globular protein enzyme, a globular protein with many amino acids. So in the active site, there are amino acids. The R groups form hydrogen bonds, salt bridges, or hydrophobic interaction with the functional group on the substrate and connect the substrate to the active site. And then the combination of the enzyme and the substrate is known as the enzyme substrate complex. So that's what we said. Enzyme active site is a region within an enzyme that fits the shape of the reacting molecule called the substrate. Contains amino acid R groups that bind the substrate. Then once the reaction is completed, they release the products. So once the substrate bonds to the active site, it is called an enzyme substrate complex, ES complex. This ES complex is when the substrate is connected to the active site on the enzyme. Enzyme and the substrate form the complex, and then enzyme releases the products. Binding of a substrate occurs when it interacts with the amino acid within the active site. So this is the mechanism of catalysis through these enzymes. So this is the process. So the substrate, which is the reactant molecule, is connected to the active site, which is a location on the enzyme, through hydrogen bonds, through salt bridges, through hydrophobic interactions, and then releases. Once the reaction completes, the products are released. They are not used. So the enzyme is not used in the reaction, and it can be used over and over again because it does not participate in the reaction. Now this is an example of the enzyme process. So the hydrolysis of disaccharide sucrose. We're using this to explain the enzyme mechanism. So the ES complex, enzyme substrate here is the sucrose molecule. So the sucrose molecule is bonds to the active site on the enzyme to create an ES complex, enzyme substrate complex. Now the reaction has to start. What is the reaction? Breaking down of the disaccharide sucrose. Hydrolysis is breaking down. So now once the substrate, which is sucrose, has connected to the active site on the enzyme complex, the glycosidic bond of sucrose is in the position for hydrolysis. Now the R groups on the amino acid in the active site catalyze the hydrolysis. They let it go faster at a lower energy, producing glucose and fructose. The product structures are no longer attracted to the active site, so they are released and allow the sucrase to react with another sucrose molecule. So this is a very good explanation of how the process works using hydrolysis of sucrose as an example. So now let's review. Hydrolysis of sucrose is what's going on. Hydrolysis is breaking down of sucrose into glucose and fructose. Sucrose is a disaccharide. So now the enzyme sucrase is participating. Enzyme sucrase has an active site, and the substrate is the sucrose molecule. So the sucrose molecule connects to the active site on the enzyme sucrase, and the amino group in the active site on the enzyme sucrase catalyzes the reaction, breaking the glycosidic bond into glucose and fructose. The products are released, and sucrase, the enzyme, is available again to help another sucrose molecule hydrolyze. So that is the mechanism of an enzyme catalyzed reaction. There are two theories. This is the lock and key model. So the active site and the substrate are like lock and key. You can open a lock with only one key, so specific. So the substrate and the active site are like lock and key, very specific. So the active site has a rigid, non-flexible shape. The enzyme can bind only substrates that exactly fit into the active site, like a lock. This model did not include, so the substrate is very limited. The active site is very limited to what substrate it can fit into the active site. The next is the induced fit model. In this, the enzyme structure is flexible. The active site is more flexible, not rigid, and adjusts to the shape of the substrate. The active site will adjust in order to bind to the substrate. The range of substrate specificity now increases because the shapes can be modified to fit. Now, shape changes improve catalysis during reaction. So let us see a model. The active site, the substrate. Now, the substrate has modified to fit into the active site. This is the enzyme substrate complex. And now you have a product, and the enzyme is back for reuse. So there is your induced fit model. The substrate and the enzyme work together to get a geometrical arrangement that works for the substrate and the enzyme. And in this, the catalysis happens, the lower of the activation energy is lowered, and the reaction proceeds. So this is the newer explanation of how the reaction works, the enzyme action works, induced fit model. So this was not the lock and key model, in which the substrate and the active site were like a lock and key. You could only work with very, very limited substrates and active sites. Now, this is a very good example about enzyme action. I want you to look on page 575, please. And these are isoenzymes. What are isoenzymes? Isoenzymes are different forms of the same enzyme that catalyze the same reaction in different cells or tissues of the body. Isoenzymes are quaternary structures of proteins. Enzymes are tertiary quaternary structures of proteins with slight variations in the amino acids. Isoenzymes we are talking about. So we will discuss 5 isoenzymes of lactate dehydrogenase. So taking out a hydrogen, that's what's happening. Here, lactate has a hydrogen, pyruvate has no hydrogen. So this enzyme, lactate dehydrogenase, takes out a hydrogen. It's a conversion between lactate and pyruvate. So there are 5 enzymes of LDH that catalyze the conversion between lactate and pyruvate. So this is what we will look at. I am on page 575, and you will see here, the same is available on 575. You can see that, you can look at the book. We are looking at two polypeptide links. The H and M are the polypeptide links. You can see the 5 isoenzymes. The isoenzyme found in the heart and kidneys has 4 H components. The isoenzyme in red blood cells, heart, kidney, brain has 3 H and 1 M. So the different subunits present of the same enzyme in 5 different locations doing the same catalytic conversion of lactate to pyruvate. So a myocardial infraction may be indicated by an increase in the level of creatine kinase and lactate dehydrogenase. So these are diagnostic tools. So by measuring these values, they are used as diagnostic tools. When an MI or heart attack damages heart muscle, an increase in the level of LDH1 or H4 isoenzyme is detected in the bloodstream. You can see that it is present in the heart and kidneys. And an elevation in serum LDH5 M4 indicates liver damage or disease. An MI may be indicated by an increase in the levels of CK and LDH. So these are diagnostic tools, and this is what we discussed. Now we are going back here now. So this is 575. And again, we are looking at isoenzymes with varying subunits of the 2 polypeptide chains, H and M, which indicate presence of all these isoenzymes of LDH indicate damage to different organs of the body. So by detecting the presence of M4, you can indicate a specific situation in skeletal muscle and liver. By HM3, lung, skeletal tissue. So these are good diagnostic tools. Again, isoenzymes. Study check. An active site is a section of an enzyme or the substrate. What is an active site? An active site is a section of the enzyme. In the induced fit model, the shape of the enzyme when substrate binds stays the same, adapts to the shape of the substrate. Adapts to the shape of the substrate, and an active site is a section of the enzyme.